Hello, thank you for watching this virtual poster presentation. This is a continuing master's degree project. We will talk about anisotropic bimetallic gold-copper nanoparticles. The authors are Lourdes Bazán Díaz, Enrique Samaniego, Rubén Mendoza Cruz, and your server Stefan Mario López-López. We decide to work on these innovative structures due to their physico-chemical properties. In particular, noble metals as gold, silver, copper show high plasma resonance response. That's why we decide to synthesize bimetallic gold-copper nanoparticles and to characterize the morphologies and the plasma resonance response through ultraviolet visible spectroscopy. Moreover, the presence of two metals and salts precursors affect the synthesis as the nanoparticles grow. The morphologies were characterized through transmission electron microscopy, and the chemical composition was characterized through electron dispersion spectroscopy. The morphologies shown in this work will be compared not just by morphology or plasma resonance response, but also we will compare the catalytic activity to reduce 4-nitrophenol to 4-aminophenol. The objective of this work is to modify synthesis parameters to obtain different shapes of gold-copper nanoparticles. But how can we obtain the anisotropic structures on these nanoparticles? Well, the synthesis was done using a modified protocol from the chemical bottom-up approximation and colloidal method. I will remind you that in the colloidal method we need precursors, a solvent, a reductant agent, and a surfactant. Here we use chloroauric acid, copper chloride as precursors, and oleylamide as solvent, reductant, and surfactant. The architect of this synthesis we called is the oleylamide, thanks to its long chain, double carbon ligand, and an amino group in a mixture of 4 of our 2 metal precursors. In this synthesis, we modify such parameters, and also we compare two pathways to obtain anisotropic nanoparticles made of gold-copper. One of them is the one-pot method, in which we mixed the precursors, oleylamide, and by heating this mixture by a certain time, we will obtain a structure. The other pathway was made by acid-mediated growth, in which we also mixed the precursors and oleylamide, but add gold seeds. So these seeds will direct the growth of the nanoparticles. In the two pathways, we modify temperature, time of reaction, and ratio between the precursors, so we can obtain different morphologies. After the synthesis was done, eventually we watched the nanoparticles after the synthesis, because oleylamine will difficult the studies on these nanoparticles even through ultraviolet visible spectroscopy or transmission electron microscopy. We show here in the results the morphologies. Our control will be the gold seeds, which we will compare its catalytic activity, not only the morphology but also the catalytic activity. The second transmission electron microscopic image is a 3-arm gold-copper nanoparticle. And was made by a seed-mediated growth. Also, the third transmission electron microscopic image was done through the seed-mediated growth pathway. As we can see here, we obtain different structures, one 3-arm, another 5-fold twin nanoparticles. And the fourth image is the resultant of the pathway of the one-pot pathway. Here we obtain a nanostar made of gold copper. But as the nanostar, the 5-fold twin, the 3-arm, and the seed-mediated growth show isocaedral structure. In this electron dispersion, we index the 5-fold twin nanoparticles, and we will see that we have the planes that express the face-centered cubic atomic array. By this, we confirm that the twin nanoparticles express the 1-1-1 facet. In this image, we show the ultraviolet visible spectroscopy response. The nanostars show high response at the near-infrared region. Meanwhile, the gold seeds show a nearly common gold nanoparticle response near 520 nanometers of absorbance. And we can see that the 5-fold twin gold-copper nanoparticles and the 3-arm gold-copper nanoparticles differ its absorbance peak. That is because of their morphologies. After we characterize the morphologies and the plasma resonance response, we test them on a 4-nitrophenol degradation. We show here the gold seeds activity, the stars nanoparticles activity, and the 3-arm nanoparticles activity. We can see that the control takes 960 seconds to reduce 4-nitrophenol. Meanwhile, the other structures have good activity compared to the gold seeds. We can see that the other structures take less time to reduce the same 4-nitrophenol concentration on a solution. We have watched how the synthesis parameters modified will change the structures on the gold-copper anisotropic nanoparticles, and how this difference on shapes and size distribution will have a consequence on plasmon resonance response. And this we can see through the 4-nitrophenol degradation by time. These results indicate that these anisotropic nanoparticles can have extended applications in other areas, such as biomedicine, reduction of organic contaminants, and biocatalysis. I want to thank the authors, thank the support from grants IA 102021, PAPIT UNAM, and CONACYT scholarship. Also, acknowledgements to José Romero Ibarra, Omar Novelo Peralta for electron microscopy assistance, Ana Bobadilla for technical assistance, Adriana Cruz Tejada, Miguel Ángel Canseco Martínez, and Cristina Zorrilla Cangas for spectroscopy characterization. Thank you for watching this virtual poster presentation.